Welcome back to the deep dive. Today, we are taking a microscopic journey right into the very blueprints of life.
We really are.
And we're not just focusing on which genes are active, but which versions of those genes are being used, and, this is the key part, precisely where that's all happening inside of physical tissue.
It's such an essential shift in how we look at things. For decades, molecular biology just saw the gene as the single unit of function.
Right. One gene, one job.
Exactly. But almost every single human gene is, well, it's a master multitasker. It goes through this process, splicing and clipping, to create multiple unique versions. We call them isoforms.
And this transcript diversity is the secret sauce for complexity.
It is. It's what gives cells their identity, and it's especially high in tissues like the brain.
Okay, let's unpack this. So for years, when scientists used these amazing tools, spatial transcriptomics, to map gene activity on a slide, they just lumped all those different isoforms together at the gene level. They'd say, gene X is active right here.
Which is like reading a novel, but only getting the first word of every sentence.
You're throwing away all the nuance, all the rich information about how a cell is fine-tuning its output.
Precisely. The central problem was that we knew this diversity was there, but we had no idea how those specific versions were, spatially coordinated, how they were defining the tissue's architecture. The computational methods just couldn't handle the data.
Until now. And the tool that changes the game is the subject of the sources you shared with me. A powerful new method published in Nature Biotechnology.
That's right. It's called SPLISOSM, Spatial Isoform Statistical Modeling.
SPLISOSM.
And its whole mission is to finally detect these isoform-level patterns from spatial data, the stuff that traditional pipelines just ignore. It's basically a specialized mathematical engine built to solve three major statistical headaches.
So that's what we're going to do. We'll dive deep into how they engineered this tool to get over those hurdles, and then we'll get to the really stunning biological findings they unlocked.
In the mouse brain, the human cortex.
And even in glioblastoma. So let's start with those challenges, because you're right, if you can't solve the math, the biology stays hidden.
The first challenge the researchers really had to tackle is what they call isoform interdependence and compositional data.
Okay, hold on. Compositional data, what does that actually mean in this context?
Think of it like this. You have a fixed budget of pre-mRNA from a single gene.
Okay.
And that gene can produce, say, three different versions, isoform A, B, and C. The total count of A plus B plus C is limited by the total gene activity in that one spot.
So if the cell makes more of A,
it has to make less of B or C relative to that total. They're not independent. Their expression levels are mathematically tied together.
I see. So a standard statistical test that assumes they are independent would just fall apart.
It completely collapses. It's fundamentally what we call multivariate and compositional.
That makes perfect sense. It's like trying to predict a soccer game by only looking at one team's score. So that's problem number one. The second challenge you mentioned makes it even more complex. Data sparsity.
Right, and this is a huge one. Spatial transcriptomics data is already, notoriously sparse. You only capture a tiny fraction of the molecules that are actually present in any given spot on the slide.
And now you're trying to divide that already small pool of molecules among multiple different isoforms.
You got it. You end up with even sparser data, which means you get a lot of spots where you have literally zero counts for a specific isoform.
And if the count is zero, the ratio is what? Undefined?
Yeah.
You can't divide by zero?
Exactly. The math just breaks. And the common workarounds, like adding a tiny number just to make the calculation work.
A pseudo count.
Yeah, that just introduces bias. It can corrupt the whole picture.
Okay, and the final big hurdle, the one that plagues all spatial biology, spatial autocorrelation. I've heard it called the noise floor, but what does that really mean?
It's not just random noise. It's the tissue's actual structure confusing the math. Think about a slice of brain tissue. A cluster of inhibitory neurons is always going to be sitting right next to
other inhibitory neurons.
Right. So neighboring spots on your map are naturally going to look similar just because they contain the same cell types. This non-independence, where proximity equals similarity, it violates the core assumption of most statistical tests.
So they flag a pattern as significant when it's really just an echo of where that spot is located.
Yes, and it leads to a massive number of false positives.
So if SPLISOSM is the answer, how does it cut through this statistical jungle? What's the core technical idea?
Well, the central solution is rooted in some pretty high-level math. They use multivariate testing with non-parametric kernels. They basically reformulated the entire problem, detecting spatial patterns and differential usage as a multivariate independence test. And for that, they use something called the Hilbert-Schmidt independence criterion, or HSIC.
HSIC and kernels. That's a lot of jargon.
Oh.
You maybe give us an analogy for what a kernel does here.
Yeah, absolutely. Think of a kernel as a kind of mathematical lens. It's a lens designed specifically for complex, nonlinear relationships.
So not a straight line.
Not at all. The way an isoform ratio changes across a complicated tissue is a tangled mess. The kernel maps that mess into a feature space that we can actually measure. It transforms the tangle into a clear pattern that we can statistically assess.
And for this project, they couldn't just use an off-the-shelf lens. They had to create two custom ones, right? The first was to deal with that autocorrelation noise.
Precisely. They developed these enhanced spatial kernels. Previous methods kind of treated every little fluctuation in the spatial data as equally important.
The signal and the noise.
Yes. But SPLISOSM realized that the big, biologically meaningful patterns, like the difference between the cortex and the cerebellum, are what we call low-frequency spatial signals. The high-frequency stuff is mostly just statistical static.
So their kernel filters out the static on the line.
It prioritizes those big, low-frequency signals. It tunes the microscope to focus only on the real biological architecture.
And the second custom lens was to fix that zero UMI sparsity problem.
That's the new compositional kernel. It's their clever solution for handling those undefined ratios. They use what they call a mean replacement strategy.
So instead of throwing out the spots with zero counts.
Or adding biased pseudocounts, yeah. They mathematically substitute that undefined ratio with the gene's global average ratio across the whole tissue slice. And they prove that doing this preserves the test's validity and power.
That statistical rigor is so important. So in the end, SPLISOSM gives you 3 different tests for total gene activity, for isoform ratios, and for isoform counts. And the simulations they ran confirmed it all works. It avoids the false positives that plagued the older methods.
And those conditional tests are especially critical. They let you test the specific relationship between, say, a regulatory protein and an isoform after controlling for the fact that they just happen to be in the same neighborhood.
You control for location first.
So you know you're only detecting a true regulatory link.
Okay, so if we zoom out from the math for a second, they didn't just build a better engine. They built a very specific tool that finally lets us see these hidden spatial patterns without getting fooled. So the question becomes, where did they point this new microscope first?
They went straight for the adult mouse brain, using long-read sequencing data because it gives you the absolute best resolution for different isoforms.
And the result?
It was a dramatic validation of the tool's power. SPLISOSM detected 150 spatially variably processed genes, SVP genes, that were robustly shared across replicates. That's more than double the 61 genes that older, less sensitive methods have reported.
Here's where it gets really interesting. What were these 150 genes actually doing? Was it random?
Not at all. The functional analysis showed these genes were overwhelmingly enriched in the pathways that, that define brain activity, membrane trafficking, and synaptic signaling.
So how neurons talk to each other?
Exactly. We're talking about core components like SNAP25 and CXDP1. These are already big names in neurodegenerative diseases like Alzheimer's and Parkinson's. It's proof that the brain is relying on this kind of spatial isoform regulation for its most fundamental jobs.
And if the isoforms are spatially organized, then something has to be controlling that organization. Which brings us to the puppeteers, the RNA binding proteins, the RBPs.
Right. SPLISOSM didn't just find the isoforms, it found the RBPs that were likely regulating them. And the results prioritized all the usual suspects, the known heavy hitters in neural splicing, the QKI, RBFOX, and SILA families.
So you have the mechanism behind the architecture.
You do. The NAS case study they highlight paints a really clear picture.
Okay, tell me about DUS.
So GUS encodes a critical signaling protein, and SPLISOSM show this really striking splicing variation where exon 3 is specifically and efficiently skipped out in the midbrain and the fiber tracts.
And why does that matter?
Because it's deeply protective. We know from clinical studies that when GAS exon 3 is included in other cell types, often because of a mutation, it creates a hyperactive protein that's linked to severe myeloid cancers.
Wait, so you're saying the location controls the RBP, which then controls the splicing, which then ensures the brain produces a functional, non-cancerous version of this critical protein?
That's what the data strongly suggests. It's this massive, spatially coordinated regulatory cascade. The brain seems to use this exon skipping to protect itself in very specific regions.
And this isn't just about splicing, right? Transcript diversity is also about what happens at the tail end of the transcript.
Right, alternative polyadenylation, or what they call trend. And yes, SPLISOSM addressed this too.
And it could capture this even with the shorter read platforms like 10x Visium.
Absolutely. The analysis found 815 SVP genes with spatially variable trend usage. So the cell is simultaneously changing the recipe, the splicing, and the packaging, the 3-fit and end, and both are spatially coordinated. And they even went a step further, using near-cellular resolution imaging to confirm that a specific isoform switch in a gene called DTNBP1 was driven by very specific cell types, excitatory versus GABAergic interneurons.
So they took it from the map right down to the specific cells.
Exactly.
So we've established the mouse brain uses this incredibly detailed localized system to control things like synaptic signaling. But this kind of micromanagement, does it hold true across species? Let's talk about the human brain.
This is the critical question, right?
Yeah.
If this is all just some quirk of the mouse brain, it's interesting, but its relevance for human medicine is limited.
But if they can find the same mechanism in the human cortex...
then we've likely uncovered a truly fundamental biological organizing principle.
So, did they find it?
They did. They analyzed the human dorsolateral prefrontal cortex, the DLPFC, and SPLISOSM, found 861 SVP genes with spatial variation.
861.
And here's the kicker. 178 of those human SVP genes had mouse homologs that were also spatially variable. That's conservation proof.
And you can actually see the evolutionary pressure in the DNA sequence itself.
Yes. The specific trend regions, the little sequences that regulate the 3-rend, showed significantly higher sequence conservation than non-variable regions. Evolution has been actively preserving the exact code needed for this spatial regulation in both species.
And the functions matched up too, right? Synaptic signaling
and chaperone-mediated autophagy, a process implicated in many neurodegenerative disorders. The septinate gene is a great example of this functional conservation.
So what does septinate do?
It's a protein that's essential for cellular structure. And the spatial analysis showed that in both mouse fiber tracts and human white matter, the cells preferentially use a specific recipe that creates a shorter isoform called septinate S.
Okay, so what's the consequence of that shorter version being localized specifically to white matter?
Well, the short version is missing a crucial piece at its tail end, a C-terminal domain.
And that domain is needed for?
It's needed to interact with another protein called VMP2, which is key for secretion. And it also contains a bit of code needed for creating complex dendritic branches in neurons.
So by skipping that exon in white matter, the cell produces an isoform that's optimized for the support cells, the glial cells, not for complex neurons.
The localized splicing decision literally dictates the protein's job description in that specific area.
That level of detail is just astonishing. Okay, so let's pivot from health and conservation to disease and, well, chaos. Glioblastoma, or GBM.
Yes, and because GBMs are so heterogeneous and messy, they're a perfect test for SPLISOSM's approach, which doesn't rely on predefined cell types.
And the findings here were translational.
Highly. They saw this pervasive transcript diversity in the genes responsible for antigen presentation, the flags that a cell shows to the immune system, things like HLA, B2M, and CD74.
So what was the tumor doing with these flags, just turning them off?
Something much more subtle. It was preferentially using intron-retaining or potentially non-functional transcripts of these genes, specifically in the tumor regions.
So it's producing a broken version of the flag-making machinery.
Exactly. And if the tumor makes a flawed version of its antigen presentation system, it effectively becomes invisible to T cells. This is a direct mechanism for immune evasion.
And a potential target for new therapies. It's weaponizing alternative splicing to hide.
It really is. And they also connected this to the tumor's microenvironment.
With the FTH1 case.
The FTH1 case, yeah. FTH1 is an iron storage protein. Its job is to ramp up when oxygen is low to protect the cell from a type of death called ferroptosis.
And SPLISOSM found?
It found that the FTH1 upregulation in the hypoxic low oxygen parts of the tumor coincided perfectly with an increased expression of 3-foot truncated transcripts.
So the cell is directly linking its oxygen sensor to its iron metabolism and its survival response, all through a precise change in the transcript's tail.
Yes, and this diversity wasn't limited to just immune evasion or survival.
It was everywhere.
It was. Even in structural proteins like GFP, which had splice variants linked to how aggressively the glioma cells could migrate. Ribosomal proteins showed diversity linked to metabolic changes. In cancer, it seems the entire genetic toolkit is being spatially tuned for maximum survival and growth.
So what does this all mean? We started with the complex math of all this, and we've ended up with a really profound new framework for seeing biology.
It means that SPLISOSM has given us this robust, isoform-centric view that proves spatial transcript diversity isn't just a side effect. It's a fundamental organizing principle of tissue architecture.
The analogy you used earlier about the novel.
It's like we were only reading the first word of every sentence, and now we can read the whole thing. We can see that every musician in the orchestra is playing a slightly different, specialized version of the same melody, all coordinated by their location.
And the translational implications are just so clear. In the brain, this gives us new molecular insights into neurodegenerative diseases.
Absolutely. And if we can understand the RBPs controlling that process, we might find new drug targets.
And in cancer. Knowing that tumors are using this for immune evasion, like those broken B2M isoforms, that's a brand new target.
If we can force the tumor to produce the functional version of that protein, we might make existing immunotherapies much more effective.
You know, we often think of gene activity as just on or off, maybe high or low. But this deep dive really shows that cells in different places are using wildly different versions of the same blueprint to specialize.
And that complex regulation, the cooperation between RBPs like RBFOX and C-self, it suggests splicing isn't just one simple switch.
It's a whole console, an entire console with dimmers and faders, all coordinated spatially for a precise function.
And since we know these patterns are so highly conserved between mice and humans, it really raises a question for you to think about. What other fundamental biological functions, beyond the brain and cancer? What's happening in the liver or the kidney? What might be quietly controlled by this hidden layer of spatially organized isoform switching?
It's a whole new layer of biology, waiting for the right tools to uncover it. So next time you hear about a single gene causing a disease, remember the spatial dimension. It might not just be about the gene's activity level. It might be about the wrong version of the gene in the wrong place.